In this video, I'm going to talk about the GPCR, G-alpha-I signaling pathway, and then I'm going to use the example of heart rate in order to illustrate how this pathway, the G-alpha-I signaling pathway, can interact with the G-alpha-S signaling pathway in order to modulate a cell's activity. Let's begin by looking at the pathway. Well, previously I did talk about the G-alpha-S signaling pathway, and you'll remember that in that system, when a ligand binds to the GPCR, it induces a conformational change activating the G-alpha-S subunit, which can then diffuse away, interact with adenylate cyclase, leading to the production of cyclic AMP and activation of protein kinase A. Well, what I didn't tell you at the time was that the S in that pathway stood for stimulatory. The G-alpha-I signaling pathway, where I stands for inhibitory, will counteract this activation. Let's see how it works. G-alpha-I signaling pathways are also coupled to G-protein coupled receptors. There will be GPCRs in the membrane, and they'll be associated with a heterotrimeric G-protein, but that G-protein will have a different alpha subunit. It will have a G-alpha-I subunit. The activation is the same, that if a signal is applied to the cell and it binds to that GPCR, well then the resulting conformational change in the GPCR will induce a change in the alpha subunit such that it drops the GDP, picks up a GTP, and as soon as that happens, the alpha and the beta gamma subunits will dissociate from one another and diffuse away. The alpha subunit can go and interact with adenylate cyclase, but when it does this, what it does is to block that adenylate cyclase molecule so it can no longer associate with the G-alpha-S. If the G-alpha-S can't bind, then none of this activation will happen. We won't get cyclic AMP production, and we also won't get any of that protein kinase A being activated. Well, now that you know the basics about how the G-alpha-I signaling pathway works, I want to show you an example of a cell where this is used. The example I'm going to give you involves regulation of cardiac muscle contraction. Now, if you know something about the heart, you'll know that the heart is myogenic. It can contract on its own. The heart has a pacemaker region that will initiate action potentials that will sweep over the surface of the heart so that we get coordinated contraction of the muscle cells. But that doesn't mean there isn't a way for the body to regulate the heart rate. So, for example, the adrenal gland can release the compound epinephrine, also known as adrenaline, and that gets carried through the bloodstream. And if epinephrine comes and binds to cells in the heart, it can actually enhance cardiac activity. The heart is also innervated by both the sympathetic and the parasympathetic arms of the autonomic nervous system. These have opposing actions. The sympathetic nervous system releases norepinephrine, the parasympathetic nervous system releases the transmitter acetylcholine. And these compounds have opposing actions where norepinephrine will enhance, make stronger heart contractions, whereas acetylcholine will slow the heart rate. What we're going to see at a cellular level is that it's the G-alpha-S and the G-alpha-I signaling pathways which will mediate these changes. Well, here's a cartoon then of a heart muscle cell. In the absence of any hormonal or neural input, what will happen is that that action potential which is initiated in that pacemaker region propagates over the surface of the heart. And this wave of excitation can act on voltage-gated calcium channels in the muscle cell membranes. Those will open up, allowing calcium to come into the cell. That calcium can bind to the contractile apparatus, the actin and the myosin, and induce contraction directly. But the bigger effect is that calcium binds to something called the calcium release channel in the muscle cell sarcoplasmic reticulum, and this binding opens up the channel so that even more calcium is released from the stores in the sarcoplasmic reticulum. The cell has to then reset itself so it'll be ready for the next action potential and the next heartbeat, and it does this with the help of a calcium ATPase pump in the sarcoplasmic reticulum membrane. It's called SERCA, and what it does is to take the calcium and resequester it. Well, now let's see how the situation is different if there's input from either norepinephrine or epinephrine. Both of these compounds act by binding to what's called beta-adrenergic receptors, and beta-adrenergic receptors are G-protein coupled receptors. They are associated with the G-alpha-S signaling pathway, so that if one of those compounds binds, what will happen is we'll get activation of the G-alpha-S subunit, activation of adenylate cyclase, which will produce cyclic AMP, and then active protein kinase A. The protein kinase A has a number of targets in the cell. One of them is that voltage-gated calcium channel, and when it gets phosphorylate, it is easier to open. It stays open longer and more calcium will come into the cell. Another target is the calcium release channel in the sarcoplasmic reticulum membrane. When it's phosphorylated, it's easier to open and stays open longer, so more calcium will be released. And finally, protein kinase A can also phosphorylate a protein that's associated with SERCA. It's called phospholambin, and it's a regulator of SERCA activity. When phospholambin is not phosphorylated, it slows the activity of SERCA, but once it gets phosphorylated by protein kinase A, it'll be more active. So what does this mean for muscle contraction? Well, what it means is when the action potential arrives at that voltage-gated calcium channel and it opens up, because of the phosphorylation, even more calcium will flood in than normal. And because the calcium release channel has been phosphorylated, when it is activated, even more calcium will flood out of the SR. And taken together, the increased calcium influx, either from the extracellular space or from the sarcoplasmic reticulum, will lead to greater cross-bridge cyclings. We'll end up with the heart having more powerful contractions. What about the role of phosphorylating phospholambin in SERCA? Well, remember that I said that when phosphorylated, SERCA will act faster. What that means is that following phosphorylation, the cell will be rapidly able to resequester the calcium. And increased SERCA activity will lead to more rapid calcium cycling. We can get a faster heart rate. It can also help load the SR with extra calcium so that when the next contraction comes, there'll be more of a calcium pool to be released, so we can get increased force of contractions as well. You've probably experienced this in your own life. If you've had a fright and had an adrenaline surge, you've probably felt your heart racing and beating powerfully in your chest. This is the cellular mechanism that allowed that to happen. Well, what about the other signaling pathway? What about the G-alpha I? We can see an example of this in the modulation by acetylcholine. Heart cells have receptors for acetylcholine. They express what's called the muscarinic acetylcholine receptor, and that's also a GPCR, but this GPCR is coupled with the G-alpha I signaling pathway. So let's see what happens when the muscle cell sees acetylcholine. Well, it will activate the GPCR, and once again we'll get dissociation of the alpha and the beta and the gamma subunits, and they'll diffuse laterally through the membrane. Remember that the G-alpha I subunit can interact with the adenylate cyclase, and it will block binding by the G-alpha S subunit. That means that the pathway that would normally lead to activation of protein kinase A is shut off. If there's little or no protein kinase activity, we won't get any enhanced contraction. In this system, there's also a role for the beta and the gamma subunits. They can diffuse laterally through the membrane and associate with a protein called GERK, G-protein-gated potassium channel. This is an ion channel that allows potassium to move out of the cell. Normally it's closed, but it will be opened when it interacts with the beta and the gamma subunits, and potassium will exit the cell. This makes the cell more negative on the inside. This matters because remember, what normally happens is an action potential sweeping over the muscle cell will arrive at that voltage-gated calcium channel and open it, allowing calcium to come into the cell. Well, if the cell is more negative because the potassium is left, it means it's going to be harder to open this voltage-gated calcium channel. That's going to delay the opening of the channel, and there'll be a slower heart rate. This system is really good for illustrating how G-alpha S and G-alpha I pathways can come together and allow the body to fine-tune a physiological response.